I'm going to take that as a yes. All right. So Dr. Helmrath actually trained here with Susan Henning many years ago. So it's really nice for him to be back in his old stomping grounds. And there he really established his interest in intestinal stem cells and how they are important in adaptation and regeneration. And since then, he's moved on to human-induced pluripotent stem cell-derived intestinal organoid models, where he's really the pioneer of making these things more complex, more clinically relevant, integrating immune system, integrating enteric nervous system, incorporating mechanical stress, things like that. And his long-term goal is really to use these as therapies in the clinic for people with intestinal failure. And he's getting pretty close to that. He set up a GMP grade lab that is very well funded to make all of this GMP grade and hopefully get things into patients sometime soon. So really excited to see what you're going to show us today. Thank you, Michael.
Thank you, Heather. It's really great to be here. Does this work here? Because I'll just stand. It's really great to be back and to have spent some time with you all. This is really going to be a fun hour for me. I'm going to give you my story from my perspective. There's many different ways to give a talk. I have learned, I think, from experience to always start now with this slide. And this just represents a fraction of the people that work in my lab now. There are many, many, many. And the key is that, you know, the work I'm going to present to you today was not done by me. It was done by the many people that enable some of the visions and the thoughts that I have had. These are very talented people. And so I'll try to recognize the people's ideas and what when they go through, but realize, you know, it's a family. Dr. Henning taught me that from the beginning, that the people in your life that love you most are your family at home and at the lab. And that you really have to recognize when you build a lab that everyone's there for a different reason. And you have to understand what that reason is and make sure that they're not just accomplishing your wants, but you're actually doing what's important for you. And that as you become a leader, I think the key is culture, and that what people feel like that they want to do and what you know they have to do really is about giving them the reason what research means. And so in today's world, there's a lot of fear, but the underlying culture of why we do what we do, I think, is really strong. I have been crying for the last couple of weeks. This was my first mentor in my surgical residency that first introduced me to some of the models that I still do today. He was 66, went to sleep, didn't wake up a few weeks ago. He was due to retire in May 22nd. So this is artwork that Brad took. This is of the Tetons and of the eclipse. And this is a picture of me with him when I was in his lab, telling me to remember where I started. Which I think I am doing today, and I still do. It's I'm not going to focus too much on this, other than the fact that what we do is important. Who we do it with is really important. And at the end, I think it makes a difference. And so for me as a pediatric surgeon, I deal with babies that have diseases that result in loss of bowel. And there's different reasons it occurs, either it's congenital or acquired. We don't need to get into that. But to call that an animal model, let's say today asked me about animal model of neck. Well, there isn't an animal model of neck. There's an animal model that looks like neck, and it's a mechanism. But to understand the disease is really to understand the fact that this is developing gut that is then insulted in a human. And that those are very uniquely different things, and they're very challenging to model. And our toolbox still doesn't have the best, but the closer we get to human is the closer I think we get to answering some questions. So for me, I want to understand the diseases, but I also want to develop and understand therapies that can help these patients. And so my mission and goal in the lab has been to do that. And I was lucky at Cincinnati to have lots of resources available to me through different means. Some of them were philanthropy, and the philanthropy that they gave me to start to build what ended up being custom, really I utilized to do things that really took me out of my comfort zone, that really helped me de-risk what needs to happen to get what we do in the lab to patients. And so I saw this, and it's now structured in 3 different ways. The one way on the top is academic research, and that's overseen by Aaron, and it currently includes 41 labs from 13 divisions. And really, the driving force of custom is to make sure we have that infrastructure of academic research, because that's the foundation of everything that moves forward. But if we want to get to research that goes out to pharma and others, it needs to be done in a way that really is reproducible and quite boring to most people in NIH labs. But it needs to be done, in my way, automatic. And so to bring in someone like Magdalena Cassandra out of industry, she was at Emulate, and to build a team that can work with industry sponsors in utilizing automated sources is unique. And that group now is well over 20 and has multiple different grants. And it's all in an automated space. And again, give Magdalena, but many people in there credit. And the bottom line is, my mission was to get this into patients, so do translational stuff. So to be able to reproduce everything in the lab GMP, and to be able to develop the safety efficacy data for the FDA, and to be able to do it really is a whole team. It includes clinicians and everyone else to find the diseases. I'm not going to have time to talk about that, but I mean, these are the things that drive me that I've been working on. I'm only going to put this up there just to highlight one of the things that I'm very passionate about, which is that you need to influence the world to do what's right for your patient. It's a patient-centric world for me. So what I do has to benefit the patient. There's a lot of other interests that come into play. And so if you want to convince an outside entity that wants to make money on a cellular product, they're going to tell you right away an autologous source is going to be too expensive. That an allogenic one, the one that can be used for everyone, makes the most cost effectiveness. But each one of you is very different. And if I took my cells into each of you and think it's going to behave biologically the same, that's not true. And to do so, I'm going to have to immunosuppress you. And if you're in the end stages of life and you need to be just survive for a period of time, that might make sense. But for me, to put a cell into a baby that immunosuppresses them that doesn't match their own tissue is silly. So to come up with a strategy to make a scalable autologous cell that represents the patients is very important, and it needs automation and then a lot of funding, and those are a huge amount of things that third group is doing with a lot of outside people outside of Cincinnati. So I can talk to you about that later. What I'm going to talk to you about is this translational embryology of human organoids that came from my work when I left here and joined work with Jim Wells and Aaron Zorn. And essentially, they're developmental biologists that understand the patterning process of the whole GI tract. And essentially, what they had done, what Jim had done, was identify the protocols to take an iPSC cell and at that time derive it into proximal small bowel. Today, the patents to do everything from your mouth to your anus have been developed with our groups and are applicable widely through in vitro systems, and I will demonstrate to you kind of how I use those models to make in vivo models. I bring up this slide because it's really important that I can remember the day with Susan when the paper came out recognizing an iPSC, a stem cell from the gut was labeled by something called LGR5, and we were interested in other markers at the time. And then 2 years later, when the culture conditions to grow them, that opened up an opportunity to learn a lot of biology, and has been tremendously helpful in some respects to the field. I call these cells enteroids. It doesn't really matter if you call them organoids. They're somatic stem cells. They represent the patients at the time of their culture, and they're hugely helpful. They weren't, though, helpful for me in making animal models because they really didn't engraft. And when they did, it was very mosaic and really didn't represent a disease. And so much of my world over the last 15 years with iPSC was to compare and contrast them with these enteroids. And as you'll see at the end, the work is very synergistic, and these are just one model. And I'll highlight that at the end of the talk. Essentially, I don't know if this movie's going to run. I will. So when we want to make a tissue and put it in an animal, really what we're studying is developmental biology and wanting to prove the fact that what we created was really a luminal gut, and that gut can fully function. And so initially we did that in the kidney capsule of the kidney, and that's a routine model. Here is one where we demonstrate that we could put these into the vasculature of the mesentery, grow it, and then later come back and to hook it up into the GI tract. And essentially, to make it work, it also has to include an enteric nervous system, and we'll talk about that. And it's an immunosuppressed mice. It has to have barrier function, or else this animal's going to die because it's exposed to the aluminum. And that, by proof of principle, is the fact that, it has a barrier function, it's a functioning gut. And that's, a model of which drives a lot of things in the lab. One of those was thought up by a very talented, now going to be a PhD in the next week or two, Holly Pooling, who's worked with me in the lab for 10 years. And Holly's really wanting to help overcome the strategy. If you look on the top left of, actually it's not in the top left, that's just the mice and the rats. But if you look at what an HIO is when we transplant it, oh, here it is, it's in the bottom. That's on the kidney capsules. That little cyst on the kidney capsule is what a single HIO in the kidney looks like. And it's like up to a centimeter. It's not going to go into the luminal stream, right? And so it's good for proof of principle. So we wanted to make this long tubular structure that we could hook up to the GI tract like the movie. And so she looked at this way of putting all these spheroids into a groove, which we now call the confined culture system because of editors. And we would grow them and find out that, in fact, they mature much faster than when they were put into Matrigel. They only need 14 days versus 28. And when we engrafted them, 100% to date have engrafted, whereas about 85% of HIOs do. So vastly different. And as you see in the bottom, they are big. Here's a picture of how big they are. That's a 3-centimeter segment of an HIO after transplantation. Whereas when we just did the typical model that you see on the top, that's a very small lumen. A lot of the times it's very hard to study. Now that we have such a long structure, it really opened the opportunity for us to do more complex in vivo studies that I'll present some of that to you, which really is important for disease modeling. What we did not expect was the fact that these were just HIOs. So this is definitive endoderm that we patterned to be GI tract. And in a publication that I'm not going to talk about, in 2017, Michael Workman, working with me and Jim, placed inospheres from iPS-derived neurospheres and showed that we had a functional ENS in that graph. But as you can see here, that ENS was not anywhere near as robust as seen in human bowel. The ENS that formed in the CCS that Holly made is very similar, at least histologically here, to what we see human, and much more robust than we ever did when we added that. So we were quite excited about that. I just include this slide to just show you some of the staining that happens in the top. You're going to see that I always include, whenever I do a presentation, human intestine, because I really think you need to benchmark against that and make sure that your staining and everything is looking like it. You'll see from my studies that I don't do just a single picture. I take what Susan tells me, gives you the huge things to show that this is representative and not just the best I can show you. And this just shows you that we're able to get the different subtypes of nerves, glia, and ganglion cells that you would expect, and that they're the same as you see in human. This is a reconstruction of a CCS bowel that we grew. Again, this is just from iPSCs from a human, patterned into gut, grown in a confined space. This is small bowel. It shows you the complexity of it. It shows you the villi on top, the submucosa, the circular and longitudinal muscle, and it shows you that the nerves that are form as you would expect in the myenteric plexus, in the inner muscle and the outer muscle layers. So very much similar to what you would think of human intestine. If you gave this to a pathologist, they would think that you got it from a human. When you do a comparison and omics of the different subtypes, you see that the HIO in the middle is lacking a lot of the components that are seen from the neural aspect that are present in the CCS as compared to human. And you can look at your favorite subtypes there in your plots. I'm not going to go into this, but I will tell you that this is single nuclear. There's a lot of data sets and annotation that have occurred in the lab to try to get this type of data. It's very, very, very challenging and now very useful for us to understand the whole GI development, which has been mostly studied on the epithelial layer, but doesn't include mesenchyme and nerves, which has become very important to me. When we grow a piece of tissue, we can then take it out and put it into a chamber, and that's what Holly did. And what this shows you is that this is 8 to 10 weeks later in a mouse. We put the grafts in and we take it out and we let it equilibrate for 24 hours so it gets rid of all the anesthetic. And then at baseline, what you're seeing here is the rhythmicity of the tissue, and that's the cells of Cajal. Those are mesenchymal in origin and they fire, and they're the pacemakers of the GI tract. And what you see is that that pacemaker in CCS is much more like the human, and that it's present in HIOs with and without an ENS, but very, very attenuated. This is just quantified for you on the right. And what this is that in our first paper, when we published with Michael Workman, we had to really give a very strong chemical signal to be able to get a response. Here we can do light external field stimulation, that's EFS. And that's what you do when you study human tissue. You give a little shock, and what you're trying to do is get that neural coupling to happen. And so when you did this EFS in HEOs with the ENS, just like in our public, it didn't do anything. But in the CCS and the human, you can see the robust response. And that means you can then study that response. In a dish, you can look at that response in the face of blocking an excitatory ganglion cell, which are prototoxin, or an inhibitory one with L-name. And if you do that, you can quantitate the fact that the enteric nervous system in this graph represents what really happens in humans, which is about 60% of the bowel has excitatory ganglion cells, about 40% inhibitory. And that's really important because most of the components of the ENS dysfunction is from the lack of inhibition, not the excitation. And most models today of all the nerves that have been transplanted are only excitatory. They haven't been able to do that with nitronergic. So this is uniquely different. If I can get the next slide. I need to go here. All right. So I threw this slide in just briefly. This is older work before that, where we grew an HIO in the mesentery. This isn't from CCS, and we sewed it to the lumen of the bowel. And you can see the human tissue staining differently than that of the mouse. And the reason I added this was just some of the conversations we had today about microbiome. And that I'm not a microbiome guy, but obviously microbiome is a key aspect of it. And what this just showed was some of the sequencing that we did of the proximal and distal gut and what's in the graft. And as you can see, there's similarities, but there's also differences that exist. And so the tissue itself that's human is actually influencing different microbiome to grow within the same milieu as the GI tract of the mouse. And so it just speaks strongly to the epithelial response and studying microbiome. And it also tells you that the tissue that we're making is capable of withstanding microbiome. Because the CCS can make such a big graft, it allows me to be more complicated. And here what I show you is a graft of CCS that's been placed now in a rat because it gets to be too big. I can't do this in a mouse. And I chose to actually sew it to the end of the cecum because size matching makes it very equal. And what you see on the bottom right is 10 weeks after it's been tied in, the stool that's in the middle. And the reason the stool looks so healthy, whereas the other ones had concretions, is that it's constantly contracting. And so it's filling and emptying constantly because it has an intact nervous system. And so it's a nice way of being able to expose human tissue to constant luminal fecal stream of the rat. Clearly, we talked about this model and how it could be used. I mean, what's missing in this model is an immune system. And I'm not showing you some of our work on making an immune system, but when we add an immune system to this rat, I really think these open the doors to IBD and other enteric diseases where you need an immune system and can function. So we're quite excited about this animal model. Lastly, What this shows you is that when you tied in the graft, the ENS markedly continues to mature. And that's what happens in humans. The GI tract in a baby is not well formed. It actually, if you can ask any mommy, the first year of life, there's a lot of colic because you're developing the ENS. That ENS happens over the first few years of life. And this just shows you that maturation that happened when the CCS compared to the non-tied in CCS has much greater contraction. And if you just look at neuromuscular coupling here with scopolamine, which links the signaling of the nerve to the contraction of the muscle, you can see just how greater the muscle is contracting after it's been tied in to just show the maturation effect of luminal exposure. So this paper is under review right now. I don't know when, it should be out soon, but obviously it speaks in the dogma of the ENS developing field, and people want to know a lot about where these cells come from. So if you think my life is easy, it's not. It's just you have to address critics, and it's just an ongoing thing. But your story is real, it's reproducible, it's going to come out. This just shows you that when we do the same thing for the colon, it makes the colon. As Heather will tell you, we can make colon, but the HCO, which we call it, never really forms a very big graft, and it doesn't form muscle. With the CCS, it totally forms a longitudinal circular muscle, and you can see how much robust the mucosa is. And if you look at the stomach, you can just see how robust that is. One of the other colleagues that worked in the Wells lab showed you have to have nerves integrated to the HGO just to get to engraft. This doesn't have anything. So it speaks to the fact that I call it bio-inspiring, and the CCS is able to make here a very foveolated stomach. But it really was obvious to me that these strategies to develop weren't ever going to be able to be done in a human. To place something and grow it and sow it in later is feasible, not practical. And what's more practical would be to be able to use a cellular therapy to heal an injury. Here, I depict an ulcer in the GI tract that hasn't healed with the concept of maybe administering fragments from our tissue that if they were to engraft and regenerate, you could repair non-healing conditions here in the GI tract. And so that largely led to us trying to come up with a clinical strategy to how that could be done. This paper actually was published. It was published in Cell Stem Cell late last year, but I'm just going to show it to you just briefly. This again is representing work that Holly did with a lot of others. And again, in the lab, we grow the HIO organoids, and they usually get transplanted 28 days. We have to fragment them to get in. Others have published the similar mechanisms, and it's under current trial right now in Japan to use enteroids that I call to be fragmented and to be put into the colon of patients with ulcerative colitis. So we chose to use that as a control to just compare head to head what the differences were from a somatic enteroid put into our graft versus fragments. We basically quantitated the number of cells so that they were the same. So understanding that the enteroids are all epithelial, it heavily enriched for them from an epithelial standpoint, where ours had mesenchyme. One of the things you can see in the baseline is that after injury, and I didn't tell you how I injure this is from warm EDTA and a brush. And so essentially it's just denuding the epithelium of I lost a slide. Denuding the epithelium of the small bowel. And then we come back later and we put in the lumen the fragmented product. This just looks at it 7 days later. And as we talked about earlier today, restitution of the GI tract is very, very, very fast, very good. And if we do nothing, there'll be an epithelial barrier on it. That's why the animal's not dead. If you look in the animals that got the HIOs, that got the enteroids that were from humans, you can see that the KU80 staining is there. It's just not very present very often. And on the bottom right, that's an HIO. And what you'll see is epithelial structures where there's robust signaling. But look at the mesenchyme. Everything brown is human. And those cells are rapidly migrating within 7 days. And if you look So now they're backwards. So here's the model. I should have looked. The model is we divide the bowel, we bring out a Y, we sew a connection, we damage the heck out of it with warm EDTA, and then in the lumen we place it. I tie off the end of it with a chromic stitch that dissolves in 3 days, and so it's exposed to luminal nutrient for above. So it allows luminal exposure, and this just shows you the H&E of how bad it is damaged in the EM before we put it in. So this is what happens 10 weeks later. If it's green, it's human, it's GFP. The top is the human. You can see it's been quantitated. If you look on the right, when we see engraftment in the HIO, the epithelium is all black. It's all contiguous. It's all human. If you look in the muscle, you can really see the longitudinal circular muscles are aligned with brown cells that are human and blue cells that are rat. And they're completely together. It is hard to find engraftment when you just use interoids, but it's there. And this shows you at the same frequency which has been published. And when you stain for it, it's mosaic. And so it's never the crypts and the villi. And so clearly they are there, but they're not there to the same extent. And so the reason for that's not 100% clear. I'll kind of allude to that at the end of the talk about things that conditions media and growing things. I think in vitro may be contributing, and some of the ways I think we can overcome that. So I'm going to transition to another talk. This is from Simon. Simon is one of the lineages of French people that came to me from Lorianne that was here, but she's driving home, so hi, Lorianne. Simon is great. He came to my lab to try to develop a cancer model, and he left doing this because cancer is hard. This was fun. What I wanted to do was I wanted to separate the endoderm from the mesenchyme so that we could start to put them back together and to look at mesenchymal factors that contribute to development and growth, and also to look at the second products. Because remember, as I told you at the beginning, the HIOs engraft very well, but the rest of them, colon, stomach, esophagus, not as well or not at all. And we wanted to understand why there was something in that, what we thought mesenchyme, that was uniquely beneficial in HIOs, so we could try to mimic that in other tissues. And so by separating and putting back, we could study it. Having different GFPs and reds, we can recombine, and as you can see by the Christmas imaging, it's faithful that when it comes from green, it stays green. When it's red, it stays red. And so this slide just shows you what looks like when we make a regular HIO and the different stainings that we have for the interendocrine cells, the secretory goblet cells, stem cell marker, OMF14. Essentially what you're seeing is that when we take them and put them back together, they develop just the same, and they do it very faithfully. So the first question that we had was, can we take the mesenchyme from an HIO and put it with the endoderm from either the stomach, the colon, or the esophagus? I didn't tell you much about esophagus yet, but I'll show you a picture of esophagus, which also equally doesn't engraft. And in fact, what this is showing you is that the mesenchyme from this proximal small bowel HIO, if done at 18 days, earlier it does mispattern, it keeps the regional identity of the epithelial tissue, and it engrafts at the same 80–90% rate. And when it does alone, it completely regenerates the tissue. So for example, the esophagus here is a very squamated with basal layer, different layers of the squamous epithelium, which matches what you'd expect in the human esophagus. And this is an in vivo model. The stomach, we had, as I said, previously demonstrated that with recombination, we can do this. This is only adding mesenchyme from the HIO, no nerve, and completely making it foveated. And then obviously the colon becomes very robust colon. So this was exciting. And it said, okay, what is it about that mesenchyme? How can we learn it? So the experiment here is to make definitive endoderm out of the same pool, and then to subset it out in the same batches to becoming either patterned in esophagus, stomach, small bowel, or colon. And so they're all with the same batch rate. And then we want to be able to look at engraftment. And just as I told you, the ones that didn't engraft didn't. The HIOs in this experiment did 50% of the time, colon, 100% of the time, colon, 50% of the time. So now it allows us to do this remixing. I showed you that first data. This just recapitulates that data, saying that the mesenchyme from HIOs is capable of supporting the others in this model. And this just basically shows the size. One of the things that Simon did in this experiment was he made sure that he calculated the amount of mesenchyme that's combined. Most of these other structures don't make as much mesenchyme as the HIO. And so by just taking the mesenchyme alone from HCO and enriching it to the same extent that it is in HIO, he was able to show that he's getting these to engraft at 100% rate. So he improved the engraftment. But if you look at the histology here, he did not improve well, this is the colon. So he didn't improve on the epithelium on another slide. You see where I'm at here. This is the stomach, where it was clear to see that when you enrich the mesenchyme to the same extent of HIO, you can get 100% of them to engraft. But when you do, you get that structure in the top where it's essentially not looking anything like colon. So you get an engraftment. You don't get the further development of muscle nor of the epithelium that you're typically used to doing. The control on the right is an HGO that has the mesenchyme from HIO as a comparative in that example. So it supports the fact that extra mesenchyme is helpful for engraftment, but not full differentiation of the tissue. And this just is more of the same. This is the esophagus. Again, we can get it to engraft, but we can't get it to differentiate. So we knew that the mesenchyme numbers were only partly correcting it. So we went back and looked at all of the omics that we got from the different ones. And the idea here was that we were going to come up with some magical, mesenchymal cell type that wasn't present or is in low expression. And I have to tell you, that's not what we found. They're very similar mesenchymes. These are annotated, but it really didn't answer the question. But what was very striking to us, if you look at the picture in the middle, was that the HIOs very much had that red endothelial marking. And we stained for it on the bottom with CD31 and HIOs, and it's largely lacking in HCOs, HGOs, and HEOs. And so Simon took the hypothesis that you need to have that endothelial population to potentially, not just to get it to engraft, but to get it to differentiate. And so the strategy that he took here was when he isolated the HIO mesenchyme from the small bowel, all he did was isolate, bead sort the CD31 population. And the only thing he gave back to the enriched mesenchymes from the other colon, stomach, or small bowel was the GFP labeled CD31. And what this shows you here is that when you recombine it with the control HIO mesenchyme, that it engrafts, like I told you. And in the bottom, it shows you that in culture here, that the CD31 cells were able to engraft within the in vitro organoids. And that when we transplanted them, they rescued. And so just that CD31 population was able to get the cells to engraft and to grow. Here I'm showing you, oh, I should be reading my slides. Susan's going to yell at me. So here I did the first experiment, which she would have told me, is I depleted the CD31 cells in HIOs. So when you deplete the CD31 cells in HIO, you significantly attenuate the organoid graft and its engraftment and its ability to form. So by removing the CD31s, you lose the phenotype in the proximal small bowel. If you enrich, as I was just saying, you can rescue them. And so here's an example of an esophagus and of a stomach that shows you that with the mesenchyme enriched with the CD31 cells, this is not a great picture of esophagus, but it's the one I have. And here's the one of the stomach, where you see a foveolated stomach on the right with the addition. And importantly, in the graft, if you look after engraftment, the green fluorescence is all the CD31 cells, which they are representing the vasculature of the graft. And so the CD31 cells make the vasculature that support engraftment. And I would predict that the blood supply that are bringing all the humoral factors that we don't know all of are very key to this fact that these are differentiating. And we just haven't learned how to reproduce that in vitro. So we said we would stop and start with enteroids. So this is a picture of what happens when you take a biopsy from a human and you grow it in your dish as an enteroid and you combine it with mesenchyme. It will engraft. This is in the kidney capsule, but it doesn't make the crypts and villi that we're used to seeing. It will make muscle, but it really doesn't really make the gut that I want. So I've always been frustrated with this because, you know, my interpretation initially was that it's the media that's probably not allowing a population to grow, or it's the fetal state of our tissue in iPSCs. But I will tell you that when we do a transplantation of NHL and do a biopsy and do the same, it looks just like this as well. So there's something that was unique and we want to learn what that is. One of the things Simon did here was showing you that when we do get these enteroids to engraft, that this epithelium does exist and it has the markers. This is for a publication that's going to come out. Again, I'm not excited about this. It just shows you that it's got the capability. And this is the best representation of a chromogranin cell, a goblet cell, an absorptive enterocyte, and a Panna cell. So it shows you that it can do it. But what was really, what this shows you is that the muscle does form. So again, I told you the mesenchyme transplanted alone doesn't do anything. It has to have an endoderm. So even if that endoderm is an enteroid, you'll make a circular and longitudinal muscle. So it's supporting that. But the muscle or whatever is not supporting the full maturation of the endoderm in that model. So one of the cool things about having a lab like mine is that they have the willingness to just do things. And this was come up with by Nambi in the lab, where instead of taking a biopsy that you typically do and you isolate and you put it into intesticult or whatever in your matrigel bubble and do what everyone has done forever, we take it and put it directly into mesenchyme. So it doesn't need to see any media. And in doing that, it grows and it does just fine. You can later isolate it and grow it in an enteroid state and passage it. But the cool thing is if you expose it directly to mesenchyme and you transplant it, these are 4 different patients. What you see is complete salvage of either the colon or the small bowel. In these examples. And so it tells me that when we biopsy from a human and we want to make in vivo models from it, and you put it directly into intesticles or any other media that you have, you lose the ability to form a full epithelium. And I don't know why, if it's an FGF or what's missing, but you can overcome that by just directly placing it in. And so this just opens opportunities to model with exposed somatic cells in all the GI tract models that I've shown you. And so these are other studies that we're excited about that we're moving forward. So, I mean, this is my summary slide. And I think, you know, in general, you know, what I want to highlight is just the importance of the mesenchyme. I think that that they're very key for in vivo models. They're also going to be really important for ex vivo models or in vitro organoid on the chips. If you're going to make something that mimics the human gut, you want to have a product that can actually do it. And many of the products that are being used for different fluidics, I don't think even have that capability. People think that, you know, an endothelial cell, a UVEC, or a fibroblast is mesenchyme. It's not. And so I don't know how it's ever going to form the crypts and villi if it can't do it in these models. So I think, you know, these things feed off of each other. I think understanding how to take advantage of the clinical protocols and why engraftment needs mesenchyme. And if you want to use a patient's own cells clinically, that's fantastic because you don't have to reprogram them. And so like the Japanese are doing, but you might need to have some mesenchyme in there if you want to get engraftment. And a mesenchymal component, albeit maybe reprogram doesn't have the proliferative aspects that the epithelium has. And from a safety efficacy, it may actually be one of the ways that we want to treat. So, I mean, it opens up a lot of translational opportunities. So with that, I'll end. I hope I didn't go over time. And as you can see, good questions make me smile.
Questions?
Hey there, Joseph Burkloff. Very cool talk. I'm curious about the need for the mesenchyme Do you know if that's because you're putting human cells into a different species, so it just needs to have human mesenchyme below it, such that if you're trying to transplant into a human, would it need that mesenchyme, or would it already have human mesenchyme below it?
So so if you have exposed mesenchyme, it can act as a surrogate, right? So, some of the earlier studies that I learned when I was in Dr. Henning's lab from Catherine Booth and others that looked at fetal tissue chopped up and put into different places. A lot of the stuff that we have seen here has been shown in those studies in the 80s and the 90s. And so the mesenchyme, without mesenchyme, it will not engraft in an isolated ectopic spot. That mesenchyme has to be there. If you expose mesenchyme as I did with the enteroids from damaged, there is no mesenchyme in those enteroids, but the mesenchyme of the animal is exposed, and then it probably supports a limited amount of engraftment, would be my bet. What those factors are very hard to know. But there are lots of factors. We talked today, a couple of you, about, what is it in the mesenchyme that supports engraftment and the role of nerves and all the things that we're studying. And why the fact that endoderm and mesenchyme have to be together to engraft is a very true statement. Independently, they won't. So it's not that this mesenchyme and the endoderm, these cells are giving signals to each other. And when they go in, they're not one sheet. They're separating, and the cells are being told what to do. But those signals initially come from the interaction of the mesenchyme and the endoderm, which I think is a really key point. Susan.
What, Michael? Oh, I'm Susan Henning, sorry. A technical point, when you do your EDTA model, because I think we tried that all the way back in Baylor
We did.
as well. Are you just putting your enteroids or your HIOs just into the lumen, or are you injecting them? Because you've previously injected them
Yeah,
as well.
so we don't inject. When you inject them, they make cysts. We put them in the lumen, and we'll deliver them in the lumen. What I didn't show you is we've worked with engineers at Georgia Tech, and we reconstitute them up into a matrix of hydrogel that, when exposed to UV light, polymerizes. And so in the large animal models, when we apply it, it's going to get lost in the luminal stream. So all you need to do is apply it, and wherever you show UV light, it sticks like glue. And then the cells migrate out over the next 24 to 48 hours. So the delivery in the human is using it's an 8-mer now of a collaborator. We use that in some of the papers in the picture there. It's just saline tied off. So it's a sausage. And the cells just have the competitiveness of whoever falls against to get in. Most do not. The fact that I put the chromic in there is that if you don't, it degenerates over time, and a pressure on a lumen, it doesn't regenerate an epithelium. So the fact that it opens, it means the secretions can drain, and it also means that the luminal content that can signal the maturation come back in. And so those are all things that we learned. And so I just use a stitched chromic. It's a catgut that dissolves in a couple of days. And we know by 7 days, because we haven't looked earlier, all of them are open by that time. And I will tell you, every model I've used in my life, I probably first did in Dr. Henning's lab. And even though they failed, they eventually had a role. I learned the kidney capsule from the pancreas people, but I mean, models themselves are very useful. It's just the cells that you put in have to have the ability, and the cells we were using at first just didn't have the ability to do what I wanted to do. Yeah.
Yeah, Gonzalez, NC State. Do you think that it might matter what injury model you're actually looking at? So in some injuries, it is epithelial loss focus, and then others, probably with your intestinal failure patients, you're seeing more of like a fibrosis, right? And so more, including the mesenchyme. So have you thought about potentially testing these with the varying models as to whether it's more of an epithelial based type of repair versus if you're needing to basically recreate the entire mucosa?
So, I mean, there is gradation of damage. You have to expose mesenchyme. So the underlying condition, if it doesn't expose mesenchyme, it's going to be hard to get it to engraft. So I didn't tell you the clinical FDA proposal is to use it in exfoliative rejection after transplantation because they slough their epithelium. If you have a short gut kid that has an attacked mesentery, mucosa, how am I going to get cells in? That I don't know. I don't have the answer without doing damage. And you don't want damage to get something in. Is damage giving signals? Probably. So those signals have to probably be there to help them migrate. So all these things have to be figured out. You know, the interesting thing is that every therapy to date is injecting. We're not injecting, we're putting it in the lumen, which means that most of the cells don't engraft, they get sloughed off. So when we're looking at off-target effect and safety and efficacy data, it looks very positive because we can't even get a tumor to form. When we put iPSCs that unpattern was supposed to make teratomas, they won't engraft because they don't have mesenchyme. So we put 5 million of these things in, they'll inject them in the subQ and they have tumors all over them. We put them in the lumen and we can't get a tumor. So you're asking, how do we get it into other areas that aren't damaged? And the answer to the question is I don't know. I think there has to be a damaged signal and there has to be exposure to mesenchyme.
And actually brought up another question,
Hmm.
sorry. And that is the amount of time. So you said that most of, you obviously have some type of matrix which your cells are in, but if you're putting it into intestine that is progressively motile, in their studies, you've kept it into an enclosed loop. Do you know about how long it takes for that engraftment to occur?
So we know the cells are in there. We showed you 7-day data. And they rapidly proliferate and migrate. So you get more and more and more. If you get earlier, it's just a needle in a haystack question. The fact that the matrigel can be tuned, it's a hydrogel, can be tuned, we can get it as long. We chose 48 hours because longer we didn't want to obstruct the lumen. And it's in there as at that time. When we do other parts, so we're going to do esophagus as the next, because esophageal strictures are very morbid and you're dilating them, you're exposing the mesenchyme with dilation. You can put the cells of the patient in that will revascularize. They're also very anti-inflammatory. So instead of these recurring strictures, any stricture in a lumen, whether it be a ureter, a biliary duct, you can imagine therapies, if you can do that, can be a treatment at the time of dilation. And I see that's where it's going to be the most clinically applicable.
So first, I'm going to read off Nathan's question, and then we'll go to you, Ian. Nathan asks, how difficult is it to successfully stitch mouse intestines together?
It's harder than rat. So a rat's easy. It's big. I mean, they're 250 grams. So if you can tie your shoes, you can operate on a rat. A mouse, you really probably need skills. And I don't think that most surgeons in my lab take about 6 months to learn how to do it and keep the mice alive. So I think it's a skill set that's not applicable to most people. I think rats are. We've trained the world on kidney capsule transplants and anyone can do it. So kidney capsule, to put it in the mesentery, I didn't tell you, we just drop it there and put a little bit of glue. Anyone can do it. It's very easy to do. There are a lot of techniques, and we've published papers on the techniques about how that is and where to place it if that helps you. But the sew on mouse bowel is something that I think is probably a unique skill of a surgeon. And if you're good, you can do it, but it does take time. And it's all done under a microscope.
Ian, if you want to unmute, we can hear you here at home or here.
Hi, can you guys hear me?
Yeah.
Oh, great, great. And I appreciate the very interesting, compelling presentation. I was wondering if you've looked at the function of these transplanted cells, I guess, particularly from an absorptive standpoint. Do you see any evidence that they are, making chylomicrons or performing in any of the normal nutrient absorptive processes? And I guess Heather's in the audience, and she's much better at these hormones than I am, but I'm pretty sure there's some EEC expressed hormones, or they might be in the stomach, things like elastin, I believe is the name of one, some hormones that are found in humans but not in mice. Are you seeing those in the mice that you've transplanted human cells into?
Yeah, so great question. So there's not a lot of things that are unique that are human derived different than the mouse. One of them is some of the fats and the stuff. And Noah Schroyer and has been working with me to try to figure out, the things that are present after the transplantation and the chylomicrons, which are very different. So circulating chylomicrons from human are different than mice. And we can see them, it's just not a lot of them. The data is not anywhere clear. When we take these and put them into a chamber bath, they absolutely can digest and take up sugars, break down proteins, and translocate fats as you will. And so, yeah, we've absolutely studied that. The question is it does, the reviewers want models that are like short gut that says I can salvage it with that. My rationale for that preclinical study is to get data for the FDA. To try to model a human condition in an animal is quite challenging. The question is an important one. How well does it absorb and digest and stuff? And to the extent that I can show you that the animals thrive and the fact that they can take it up in an X chamber bath, that's the only data that I have. I don't have an in vivo way of absolutely convincing you that GI tissue it is. I will tell you, if you look at the picture that I had before, the white cells that are in the lumen, that's chyle. And so I did studies where I made the chyle oil red. Right there. All that white that you see here, that's all chyle. And the lumens of the villi are completely full of the fats. And so the lacteals are full. So, I mean, I don't know if that tells you they're digesting fat, but their lacteals within their villi are full of chyle. That's probably the closest slide I can give you to answer your question.
That's fairly convincing. Thank you.
Hey, I'd like to go back to the first story for the CCS model. So they're becoming much more mature, much more physiologically relevant. Is it purely that they're physically constrained, or are they in any kind of different matrix, any kind of different
Nothing.
media?
So we bioprint the confined dish to the size that makes it modifiable. Let me go to the actual dish that Holly makes. And so these are the pictures of the dish here. And within 6 days, it's a fibrous tube that we take out and we pin and we put it like in a matrigel dome. So they stay only in the dish for 6 days. When she took them out like 28 days, they look like crud when they transplanted. We went back to 14 days and they fully engraft. An HIO at 14 days will not engraft. It engrafts between like 28 and 35 days. And like the Spence lab has done it longer. He adds conditions for for the epithelium. We don't. The only thing we have in our media after patterning is just EGF and the supporting media. So they're in that same media here. There is matrigel that's coating, but you see most of their contacts. So they are, the tray is coated in matrigel. We have tried this in the fine hydrogels that we're using for GMP, and it's not completely replicated. They don't stick together enough. I think it has to do a lot with the tension and the force of the matrigel, which we need to tune to try to get these cells to become more integrated as a tube. But by 6 days, this comes right out like a piece of spaghetti. And then they pin. If you don't pin them, they'll curl up.
So then for why they act so different, do you think it's just a stiffness effect, or what actual physical effect is driving
I
that?
think the fact that cells are interacting with each other is a really important cue, that the tension and the force, and that what happens in matrigel is that the mesenchyme is like it's holding on to the proteins. And so the fuzziness you see in our pictures is the mesenchyme grabbing the laminins and stuff, and they want something to attach to. If they got a cell next to them, they're hugging that cell, and they're getting the signals and the growth factors and stuff to differentiate into pattern that is highly regulated. We try to reproduce that by adding factors to, but these cells are making it, right? These cells are further telling each other what to do. So what it is that they're making, I don't quite know. We certainly have a lot of omics that we're looking at. We have omics on the CCS from every day, 0, 1, 2, 3, 4, and now we're doing it every 12 hours and trying to annotate when different populations are coming up. So it's a tremendously large data set. It's still very difficult because those populations haven't been annotated. And so it's challenging. And when you ask, is it a nerve? Is it muscle? Is it endoderm? As I will have shared with Heather and others, it's not that simple. And these guys didn't read the textbook, so they're not following the patterns that you would expect from the literature.
That's an amazing model. Thank you.
If no one else does. So in the CCS model, it's the same starting population that makes everything.
Yep.
Do you make lymphatics
What do you mean in the
after you transplant?
So that's a good question. There's not a human lymphatic marker that we have, but we see lymphatics. So do I think I have lymphatics? I do, but you gotta prove that it didn't come from the host. And so when you try to do human staining and co-staining of lice, I think it's Ly1, but it stains both lymphatics of the mouse and the rat and the human. So it stains everything. We don't have a unique human lymphatic marker. We have lymphatics that are in the graft, and everything else in the graft is human. So I would assume it's human, but that doesn't define it. So unlike
It's a human nuclear marker.
So we
Sure
did
is.
that. So,
Yeah.
and I didn't tell you this, but the blood vessels that form in HIOs, even though endothelial cells are in HIOs, 100% of the blood vessels in the graft are from the mouse. They're MECA32 positive. So they never form blood vessels until you give the nerves. When you give the nerves, half of the vessels in the graft are human and half of them are mouse. So I know all vessels in that are not. So to say lymphatics is to have that same MECA32 versus the human staining, where you can see that's a human blood vessel, and that they connect. I can't do that with lymphatics. I just can say it's lymphatic. And I can say it's human because the graft is human, and I do see human markers, but as you know from human markers, it's not a pretty line of this is human and this is mouse. It's not 100% popular. So yeah, it's one of my to-dos that I'd love to be able to prove the lymphatics, but that's what I got.
I see hand raised online. Jadon?
Yeah, hi, great talk. Thank you. Could you go to the slide about the co-culture of the patient-derived enteroids with the mesenchyme? I just want to make sure I understood you correctly. So you can take your somatic enteroids, which are epithelial, right? And then you can mix them with the mesenchyme that you've taken from another patient's HIO. And
Yeah,
then,
so
and that allows it to engraft, but not as well as the original HIO.
can you see the slides?
Yeah.
So this is the example of the typical enteroids that are in the lab all the time that are being passaged, and the controls, whether they came from an HIO, they had to if they're GFP because they're GFP humans, or they're just regular humans. But we've done the experiment many times and show that we can get them to engraft. But what looks like in the bottom right is what we typically see, which is just an epithelium. And there might be areas that you call crypts and villi. And Simon did his best job. This is for the paper to try to prove that he was making. But this to me is not exciting. What was exciting is that now we've done this over, I think, 12 times, 12 different patients. This is just 4. Where whenever we take a biopsy now and we bring it back to the lab and we isolate the crypts that you typically do to make an enteroid culture, you put some directly into mesenchyme and you just plate them out, and then they'll form, cysts, and they'll grow, and you can split them out at 7 days, and at 14 days, if you want, you can re-isolate the endoderm from them separate from the mesenchyme, and then we passage those, and those are the ones we're studying. And we're hopeful that they maintain the same biology that happens when we isolate endoderm from an HIO in vitro. That endoderm, we also have to passage in intestinal cult, and we can do that multiple times, 5 passages, but when we recombine it, it still forms this exact same thing. And so the hope is we can start to generate human lines that don't even need the mesenchyme long term, that when we combine, they can still form this. And the question is does the biology influence, or does it change the biology of an enteroid to have that capability? And I don't know that question, but I know if you want to make a model that looks at a complex GI, especially like an in vitro on an organ chip model, that you want to model something, I would use an enteroid that can do what you see on the bottom and not one that can just make a cyst, because I think your likelihood is you're going to get a better model.
Oh, thank you. That was really cool. Yeah.
Ian, you want to ask another?
Yes, I just have a follow-up to Jackson's question. Is this effect unique to your HIO produced mesenchyme? Like if you take mesenchyme from a donor, a human donor, and grow it and put crypts straight onto that, do you get the same boost in regeneration?
It will die like a smelt. Yeah, so mesenchyme that you get from human does not culture well. It becomes fibroblast. It doesn't support engraftment. It does not have the characteristics. And we aren't able to passage mesenchyme without it losing its developmental structure. So unlike endoderm that we can passage over and over again, mesenchyme is going through a temporal spatial period of development that supports this. So we can freeze it down as a product that we do so we can thaw it, but you can't keep mesenchyme in addition, expand it, and then just apply it. It is a very temporal and spatial effect of the mesenchyme during development that we get roughly at 18 to 21 days in an HIO of the mesenchyme's ability to do this. So it is a developmental effect of HIO mesenchyme from iPSCs, and I don't have any other examples of where anything else works like this.
Okay, thank you for clarifying.
Thank you very much.
So obviously I should have looked at my slides.
So